There we go. Hi. Okay, so let's talk about this one figure. This is figure one from the paper, and I'm going to draw a little bit on what I was able to read in the supplementary data. So you can use either the published version of the paper from Science or the version on bioRxiv. Those are both fine for the paper. I think for most of the supplementary information, you have to go to the online version. And if you click on the Science link, even if you're not able to get to the article because of where you are or if they haven't made it publicly available, you should be able to get to the supplementary information. They normally make that public as just part of publishing it, and that's always going to be free. So there we go. So, all right, what are they trying to do here? What they're trying to do is take the antibodies from a person, make tons of them, but just the ones that work really well, these neutralizing antibodies, and then just sort of ramp them up. And over the course of a week and a half, they want to go from antibodies that are in somebody, that are being produced by their cells, to antibodies that you could inject into anybody. And potentially, this would be a really great therapeutic. This is a paper that I feel like this is going to be a fairly important paper down the road. But we'll see. I mean, hard to predict these things, I guess. So I want to go through this figure because once you understand this figure, all the rest of it is just small details and you'll have the basic concept, and I think we'll be good. All right, so step one, they take a donor. So this is like, remember when Tom Hanks said he was donating his coronavirus-educated blood or whatever to try and make a new vaccine? The Hank scene? Yeah. This is going to be something like that. I can't find any connection between this group and Mr. Hanks directly. But of course, all that would be confidential, and he may have donated to another group. There's a lot of different groups in California that would be trying to do something. Okay, so inside the bloodstream of these people, they will not just have antibodies, but they'll also have some of the cells that are producing the antibodies. Those circulate around in the blood with all the other white blood cells. And this is good because we can pull these out. So an antibody-producing cell is going to be a thing called a B cell, and, well, actually, once it starts producing, you can also call it a plasma cell. These would mean the same thing in this case, as far as we're concerned. The distinction is technical and is for immunology class. So these circulating B cells, they're going to capture those. And a B cell does two things. One, it'll make antibodies, and the other thing it does is it will store a copy of the antibody it just made locked on to its own membrane. So the B cell will be covered in the antibody that it makes. It's like a store. Advertising's like, here, this is what I've got. The B cell's not really advertising, it's putting those on the surface because this is how it learns whether or not it is making useful antibodies. If it gets a signal that it's hit something properly and another cell can come along and reward it, then you've got a reason to make more antibodies, and that's the signal for this antibody-producing cell to divide. So there we go. So up here in the top corner, they've got a person with a coronavirus inside of them. Great. And this person's presumably recovered. Convalescent means they've recovered from the disease. And so they're taking out plasma from this person to see what kind of antibodies do they have. And they're presumably checking the antibodies from this person, because if they don't make very good neutralizing antibodies, then you're not going to get a good result from this entire test. But if you can start with a person who has really good antibodies, and maybe Tom Hanks didn't make good antibodies. I mean, I hate to say anything about the great man, but it's possible. Then the other thing they're going to pull out are peripheral blood mononuclear cells, which is just, let's just say, white blood cells that will stick to a dish of some sort. So this is going to include things like the B cells that are producing antibodies. And so from this, they're then going to take the spike protein. So here's our spike protein. That looks familiar. And what's this thing sticking up at the top? Here's one that's down. There's another one that's down that's hidden there on the back. And here's one that's up. That's the receptor binding domain, right? Yeah, we got this. Okay, and so they're expressing just the receptor binding domains. Neat. And then they came up with a way, and let's just assume they use technology to stick receptor binding domains onto something. There are lots of ways they could have done it. There's one way that they happen to do it. It doesn't really matter for our purposes. It's just a receptor binding domain stuck to something that you can detect. Okay, so they're got the receptor binding domains, and so what they're going to try and do is label up the cells. They're going to use some kind of detector molecule with receptor binding domains on the outside. So it's going to be like a little fake version of the virus. And this is going to stick to B cells that are making antibodies that produce, that are good, strong binders. So not all the antibodies that are bound by the receptor binding domain are going to be neutralizing antibodies. It'll have some antibodies that are kind of near misses, if you like. But you're at least going to push it because we know that something like 19 out of 20 neutralizing antibodies hit that receptor binding domain, at least from what we've seen so far in the literature. And so this is the best way to kind of hedge your bets toward finding neutralizing antibodies. Okay, and so they are then going to sort the cells, and so that's what we've got going on down here in B. This is a flow cell sorter. It's also called a fax machine, FACS, and this is a way to label cells. So you can label cells with, one color to indicate that they're B cells and another color to indicate that they have bound the receptor binding domain. So if you put a fluorescent receptor binding domain on that's green, and you put a red antibody, fluorescent antibody that'll bind to something else on B cells, then anything that binds both is going to end up in this quadrant up here, which indicates that, yeah, it's a B cell because it's got a B cell marker, and it is binding very nicely to the receptor binding domain of the spike. So it might be making a very good antibody. And so they take all those cells and they sort them out. And then they stick them one at a time, one cell in each little well of a many welled plate. Doesn't say how they did this. I would imagine it's probably not even a 96 well plate. It's probably something smaller than that. Whatever 96 times 4 is. 300 and, I don't know, 390 or something like that. 392? I don't know. Math fans, go do that. But those plates would work very nicely, and so each little cell has a little tiny home in there, and they put some nice growth media on them so that they can stay alive. And then they just take each of those cells and they open it up. And so what we're doing first is this RT reaction, and this is a way, it's called reverse transcriptase. It's a way that you can copy RNA and turn it into DNA. The reason they do that is because we can't really do anything with RNA, but we can do anything that we want with DNA. This is the idea. What are they copying? They are looking for the actual heavy and light chain gene that single cell was making. And they're going to try to pull out the pair. They're going to try to do two separate PCRs to pull out a heavy chain and pull out a light chain. And so on day 2, they're going to do the PCRs. This one's called a nested PCR. Don't worry about that. It just means a regular PCR, you would have 2 primers outside of the gene you want. For the nested PCR, you just repeat the PCR, but with 2 more primers that are just inside the first 2 that you used. And so it's a step that if you had a kind of a messy first step, this will give you a more specific second step to it. That's what they're trying to do. They don't go over exactly how they came to that, or whether they're doing it because they thought it might work or because they have to do it, but that's fine. Okay, so then you have your PCR products. You've tried to amplify heavy and light chain from each one of these cells, and yeah, you can do this from a single cell. This is a modern technique, single nucleus PCR they call it, and it's honestly a pretty powerful technique. You can see what an individual cell is making and what makes different cells behave differently. So then they push the fragments down a gel, and the pair determination is they look and see, oh, do I have a heavy chain and a light chain from this cell? And if they do, then they have determined. They have a pair. Yeah. Isn't that a fancy word for, counting to 2? All right. So Day 3. They're going to go in and they're going to put their heavy chain and their light chain, which I don't know why they call it AC instead of LC, but and also it looks like a kappa chain for some reason in there. And so they're copying these out, these pairs, and they are sticking them into plasmids. And so they're then going to try to, you stick a gene into a plasmid so you can make lots more of it. And they're putting those plasmids into E. coli, and they will just grow whatever plasmid you put in there. And so we're going from a single cell to a gene that we've copied out to a plasmid that will copy that gene and carry that gene forever. And we can do all this without knowing what the gene is. We have primers that'll bind to the beginning of a heavy chain or a light chain and the end of the same chain. And they will amplify whatever it is in the middle. So you can amplify any kind of antibody that is made. And each B cell is only producing one kind of antibody. They can't produce different kinds. Each one just decides on one kind of antibody, which is why this works as well. The reason they were using RNA back here instead of just reading out the DNA is because the RNA is going to be spliced. And so it's going to have just the exons and not the introns. If you remember back to like biology 1 ages ago. And so rather than having to read out several 100,000 nucleotides to try and pick out which parts are the exons, which is the way the genes actually look on the chromosome, they can just read out the messenger RNA that's all trimmed down and tiny and yeah, it just contains the good parts. Okay, so now they've got their heavy and light chains, and so they can express these things. They can put these things into some kind of system, and these are genes. They will just grow antibodies. And so they are then mini prepping, so they're just pulling the plasmids back out of the bacteria. You put one plasmid in, and you can get billions of copies of the plasmid out in about a day, like they've done. So at every step here, we're going from one cell and we're trying to make more and more and more copies of these things because biology is basically pretty inefficient, or molecular biology is. And so we can't really work with just one thing. We've got to work with billions or trillions of copies of one thing in order to effectively do anything with it. Okay. So then we take those DNA, we put them into cells, and presumably these are human cells, target cells of some sort that are capable of producing antibodies. So this might be a just a human B cell, something like that. And then they put these in, the cells start making antibodies, and you've got to leave it a couple days. This is the one really slow part where you can see day 5, 6, and 7, they're just showing more and more antibodies. Because, yeah, these cells, this is their job. They will produce antibodies. That's what they do. That's what makes them really cool. And so then, on day 8, they'll take the antibodies that they've got, probably first thing in the morning. So really, they've grown these antibodies for, well, they put them in day 4. So 4 to 5 to 6 to 7 to 8. Wow, yeah, that's a long time. But that's how long it takes to grow large amounts of antibodies. Antibodies are slow. This is their problem. But they're really powerful, so this is their good side. And now they're going to do a thing called a neutralization assay. And the neutralization assay you've already heard about. This is where you put in something like a virus. And you can do it with the real virus, or you can do it with the shell of the real virus, like the proteins on the outside of it. And instead of the real virus genome, instead of this being the SARS coronavirus 2 genome, you can put in a green fluorescent protein gene. And so the assay is you try to get this thing into cells. If it gets into a cell, it's going to deposit the green fluorescent protein gene, and the cell is going to start glowing green. Or there's different fluorescent proteins, different ways to detect it. But if your antibodies block this thing, if they just stick all around it, then they are not going to, it's not going to be able to start that infection. And so, yeah, no. So neutralization means cells don't get infected by your little virus. This right here is your little virus. And yeah, they're just looking for how much of this antibody do I need to put on, and will it stop a virus from getting into cells, or does it just bind to the virus and not stop it from getting into cells? Because those would be useless antibodies. So now, for each one of their individual sets. They have a heavy chain and a light chain. They can now check and see which ones are the good antibodies, which ones are most neutralizing. And they can just select those neutralizing antibodies and throw away the rest. So this is a way of basically sorting through the immune system for the things you want and just making lots of them. Only you make lots of them first, and then you just end up throwing away most of them and keeping the good ones. It's like baking 200 cookies to keep 5, something like that. And so then, now that they have their antibodies, they're going to do an ELISA as well, just to check that the antibodies still bind to the spike protein, and so that's fine. That's almost, maybe that's like a Saturday or a Sunday, and people just need to rest a little bit. Or maybe they set it up so that the Saturday or Sunday falls in here, because you don't really need to do anything on those days. The cells just grow themselves. You don't even need to watch them. Yeah. Okay. And then a neutralization test actually takes about 2 days to get done. And so that's why they're doing the ELISA on day 9, because they have a day when they have to wait. Because you put the virus in, it actually takes a while, maybe 48 hours for you to get enough fluorescence out. And they're reading out the amount of fluorescence. And so numbers that are over here are going to be, actually, no. Sorry, these are neutralizations. So this is no neutralization at the bottom. This is 100% neutralization at the top. And this is adding very little antibody, and this is adding lots of antibody. So as we add more antibody, we should get neutralization. So like for this blue antibody at the end, if I'm reading that color right, it may even be black. It's so tiny. This one's probably their best antibody. Over here at this side, the purple one, this one's pretty terrible because you got to put in that much antibody to get the same effect as that much antibody from this one. So they probably picked out the best couple from here. And then they already had these things prepared because they had prepared the antibodies actually back here. And so you probably have a whole team of people doing this. This is not a one-person job, although you could almost have one person get through it. But if you look how many names there are on the beginning of the paper, that is not how they rolled. Yeah, they did not leave this to one person. So this is fine. You can then take these antibodies and you can try and figure out, do the structure, where exactly do they bind, whatever. That's optional. But what they did then in this paper is they took little hamsters, Syrian golden hamsters, I think they are, and they gave them the virus, because these hamsters can catch coronavirus. They're a good animal model. And they also gave them the antibodies, and so some of them would have just got the virus, some would have got virus and antibodies, and if the antibodies do their job, then it will knock out the virus and the hamster will not get sick, and this is exactly what happened. So that's where we're going with this particular paper, into the world of how to, it's like panning for gold, isn't it? You start out with just all the blood from a person. You don't have to drain them completely out. You just take a blood sample because it's going to have lots of cells in there. You sort out the good cells. You take out just the right genes from those good cells. Throw the rest away. Yeah. You take those genes, you copy them, you put them into other cells that make you antibodies. You screen the antibodies, check out which ones are really, really good with multiple tests. And then you can just use these antibodies. And so it is 10 days from antibodies being inside a person and you not knowing whether they're good or bad, to the point where you can actually figure out that, yeah, these are awesome antibodies, and you've made enough of them that you can treat a hamster or a person with the antibodies, and you can protect them. So right now, this convalescent plasma, this taking antibodies from one person and putting it in another, is something that's done pretty widely, but each time it's different. You can only take so much blood out of a person, and they only have so many antibodies in that blood, and you're not putting the cells into that person. That would be like a transplant, and they would have to go on anti-rejection drugs. You're only putting in the antibodies. And the antibodies only last so long. It's a matter of days that antibodies would hang around, possibly weeks, until the last ones are gone. But they don't last all that long in the bloodstream. And this is just a super way to shortcut that entire process And if we could start to roll this out, if they could take even the antibodies from several people that make really good antibodies and just start to mass produce these, this would be a pretty excellent antiviral, I think. So that's why this is important, and this is fast. Imagine if we had neutralizing antibodies that could be given to people on the 10th day of this outbreak back in, I don't know, November or December last year. You could have started treating people instead of stumbling around and trying to figure out, where's it spreading, what's going on. Yeah, we could have had treatments right away. That's the power of this approach, and that's why I thought this was a pretty cool paper to look at. So thanks very much. That's the end of this one.